Hi, it's so nice to meet you. I'm Dr. Victoria Parikh, and I am the director of the Stanford Center for Inherited Cardiovascular Disease at Stanford School of Medicine. In our center and at expert genetic cardiomyopathy centers around the world, we are trying to understand how best to care for patients like you who have inherited or genetic cardiomyopathies. The topic that I'm going to be discussing today is gene-elusive cardiomyopathy. We've been talking a lot in our community about what having a genetic diagnosis means for a family with a heart muscle disease. But I want to talk today about what we know for folks who don't have a genetic diagnosis and what we're doing to get them closer to having one. So let's start first by talking about what a genetic diagnosis is and what it then means to be gene elusive. So as many of you know, when you see a cardiologist and you have a cardiomyopathy or a heart muscle disease, and that cardiologist does a bunch of tests to understand whether there is an acquired reason for your heart muscle disease, like blockages in your arteries that feed your heart, or exposures like chemotherapy, or for example, maybe you actually just have an athletic heart because you've done so much exercise at such high intensity in your life. Once your cardiologist has ruled out those causes of your heart disease, and especially if you have a family history of heart muscle disease or unexpected early death in your relatives, then your cardiologist may decide that the right thing for you is to get genetic testing. Now, genetic testing is a way for your cardiologist to understand whether there's a misspelling in one of the genes that's responsible for the structure of your heart and the function of the cells that make it up. Importantly, when you get genetic testing, we are sequencing every letter in the sentence that is that gene. And of course, all of those genes go together to make up a book of sentences that is your genome. We know over 100 genes that have been associated with heart muscle disease or cardiomyopathy. And so generally, we focus on sequencing those genes at really high depth to make sure that we are reading every letter in those sentences correctly. We identify different letters in those sentences that have changed from what we understand to be the reference human genome. But it turns out that we don't understand the impact of every misspelling in each one of those sentences that makes up the genes that are important for how the heart works. So when we find a misspelling that we know has either caused disease in other families or that we know enough about the biology underlying that gene to tell you that that misspelling can break that gene and cause a malfunction in the heart, then we can give a genetic diagnosis. For most people, as we're going to discuss, who have cardiomyopathy, we are unable to identify a known disease-causing variant or misspelling in their genes that can explain their heart muscle disease. There are several reasons for that, which we're going to get into in just a second. But I just wanted to take a moment here to define what gene-elusive means. What that means is that a cardiologist has decided that you may have a genetic cause of your particular cardiomyopathy or heart muscle disease. And so therefore, they have decided to send a genetic test for you. Having a genetic test that does not identify a known genetic variant that causes cardiomyopathy doesn't mean that your cardiomyopathy doesn't run in your family. It doesn't mean that it's not genetic. It just means that we don't know how to interpret the misspellings that we see on your genetic test, or that we don't know where to look in your genome to find a new gene that might be causing your family's heart muscle disease. So importantly, in some cardiomyopathies, for example, dilated cardiomyopathy and arrhythmogenic cardiomyopathy, up to 75% of folks who have a genetic test will not have a genetic diagnosis. And those are the folks that we would refer to as gene elusive. That doesn't mean, again, that they don't have a genetic cause for their disease, but it does mean that we aren't sure where to look for that genetic cause. For about 25 to 50% of other patients with cardiomyopathies, they will have a genetic diagnosis because we do know where to look and we do know how to interpret the misspellings that we find. In DCM and ARVC and ACM, as I mentioned, we have about a 25% diagnosis rate with genetic testing. And with HCM, it's about 50%. So why does having that genetic diagnosis matter? Well, it mostly matters at this time for family screening. What that means is that if you have someone in a family, this is a picture of a family that we call a kindred, that has a known disease. So let's say that this person with the red circle has hypertrophic cardiomyopathy. If we can make a genetic diagnosis for them, then we are able to test their family members to see if they carry that genetic variant that we think is causative of their disease. If that patient doesn't have a known genetic cause of disease, we can still screen their family members. We simply can't do it with their genome. Instead, we use familiar tests like echocardiograms, ECGs, and rhythm monitoring in order to be able to detect the onset of disease in their family members. So for patients with gene-elusive familial or inherited cardiomyopathy, we are still going to catch disease in their family members before it starts, as long as we can get them to a cardiologist who will be able to detect it in them early before the really feared outcomes of heart muscle disease occur. We do know some things, though, about how patients with gene-elusive cardiomyopathy do as compared to those patients who have a genetic diagnosis that explains their cardiomyopathy. This is a figure that comes from a very large collaboration that has been ongoing for over 10 years amongst at least 15 centers around the world for hypertrophic cardiomyopathy. It was led by Dr. Carolyn Ho at the Brigham. And the question she asked is, if you follow patients across their lifetime, so that's the x-axis down here, that's their age in years. If you follow them across their lifetime and they have a known pathogenic or disease-causing variant for HCM in green, or they don't in red, that if you follow them, that the folks who have a known causative variant have more heart failure and arrhythmia events than those patients who don't have a known genetic diagnosis. Now, you'll notice that there are patients here in blue. Those are patients with a variant or misspelling in a gene that we know can cause cardiomyopathy, but we don't know if that particular misspelling actually causes disease. Those are called variants of uncertain significance. You see here that those patients actually have sort of a middling outcome here. They do better than patients with a known pathogenic or causative variant, but maybe slightly worse than patients who didn't have a variant or misspelling in any of the genes we know to be associated with HCM. So this is really actually somewhat reassuring for our patients who did not receive a genetic diagnosis, even if they have one of those variants of uncertain significance or misspellings in a gene that we're not sure how it affects the function of that gene, because it means that overall, this population of patients actually does a little bit better than those with a genetic diagnosis in HCM. Similar data has also been shown when we compare patients with genetic diagnosis in dilated cardiomyopathy as compared to those inherited dilated cardiomyopathy patients who don't have a known genetic cause of their disease. It's also important to note that actually, as we mentioned, most families who are living with heart muscle disease or cardiomyopathy are living without a genetic diagnosis. This is another piece of data that came out of our collaborators in the Netherlands, showing in all 48 genes for which we have high confidence that there are variants that cause cardiomyopathy, that there are mostly patients who have variants of uncertain significance. These are in gray bars, and a very small number of patients, again, about 25%, who have known pathogenic variants in these genes. And what that means then is that we really need to understand what these variants of uncertain significance mean in order for us to do a good job in being able to provide diagnoses to patients who have these misspellings in their genes that we don't quite understand yet functionally. What that comes down to is trying to understand in the laboratory what the consequence of each one of those misspellings is. This is some ongoing research that's happening across the US, funded by the National Institutes of Health, where a bunch of different scientists and physicians, pictured down here at the left, are trying to go through systematically the genes that we know cause cardiovascular disease. This includes not only cardiomyopathy genes, but also hyperlipidemia or high cholesterol, as well as early arrhythmias without cardiomyopathies. What we're doing is going through systematically, and we're making every possible variant, every possible misspelling in that gene. What this graph shows is that on the x-axis here, on the horizontal axis, is every position in one of the genes that we're looking at. This gene is calmodulin. It causes early arrhythmias. And we're taking every possible position and we're changing the spelling of that position to a different letter. And we're seeing in single cells how that changes the function of that gene. And what that allows us to do then is to take variants like those that are reported here and to say whether or not we think those variants are actually causative in a patient or a family in which they're found. The idea of this is to be able to resolve variants of uncertain significance so that we can add genetic testing to the screening that's available to these family members. Additionally, work is being done in families with dilated cardiomyopathy who don't have a known genetic diagnosis to be able to identify that genetic diagnosis and understand the factors that lead to the onset of disease. Importantly, we know that the penetrance, meaning the likelihood of actually developing disease if you have inherited that genetic misspelling, whether we can detect it or not, is actually between 30 and 50%, depending on the type of cardiomyopathy that you have. So it's more like 30% for dilated cardiomyopathy and more like 50% for hypertrophic cardiomyopathy. In the DCM consortium, what they're doing is sequencing all of these families, trying to identify the causes of their disease by lining up their genomes and looking at what those folks who have disease in those families have in common in their genomes, which misspellings they have in common. And then they're also looking at what lifestyle factors actually change the penetrance or the probability of those patients getting cardiomyopathy in the future. You also may have heard that we have new therapies coming to light for cardiomyopathy. You have likely heard that these are gene therapies. And when you hear the word gene therapy, you might assume, as would be totally reasonable, that it wouldn't work for someone who doesn't have a known genetic cause of their disease. But what I want to tell you about today is efforts that are ongoing to use gene therapies for patients who don't have a genetic cause of disease. The way that this is done is that we still use really cool tools like CRISPR, which you've heard about. It won the Nobel Prize several years ago. We can also use other forms of modifying the way that the heart expresses certain genes or doesn't to be able to target networks of genes that work together in the heart so that regardless of what the initial genetic cause is, we're able to reverse or prevent cardiomyopathy. A couple of examples have come up with this. The first one is actually in high cholesterol. This is some data that was presented in 2021 in the journal Nature from Kiran Musunuru and his group. What they did here, and this is in primates, which are relatives of monkeys, what they did is they said, okay, we are going to we're going to use CRISPR to delete a gene that we know is bad for high cholesterol. And it's bad for high cholesterol no matter where the causative genetic variant is in a family for high cholesterol. That gene is called PCSK9. You may recognize that name if you're on a PCSK9 inhibitor, which is an injectable medication to try to reduce the activity of this particular protein, which raises cholesterol levels. What Dr. Musunuru's team showed is that they used a CRISPR base editor to delete the copies of PCSK9 in these monkeys. And what they showed is that when they did that in black down here, their overall cholesterol percentage dropped to 50% of what it was before that gene editing technology was delivered. And up here in the monkeys that were not treated with this gene editor, they actually showed that the cholesterol continued to rise over time. What this tells us is that for many genetic causes of disease, if we can find a linchpin target, we can use gene editing to get rid of that target to help anyone with a disease like cardiomyopathy, whether or not they have a precise genetic diagnosis. Very early data that was just presented at the European Society of Cardiology suggests that this may be a tractable strategy for patients with cardiomyopathy that is inherited as well. This data shows that delivering a protein inhibitor via gene therapy of one of the important negative actors in inherited cardiomyopathy called protein phosphatase 1 is safe in patients with later stage cardiomyopathy and is opening up the hope that it may actually help to reverse disease. So with that, I'd like to end by going over some take-home points. The first is that genetic diagnosis is made when a known disease-causing variant or misspelling is identified in a patient with an inherited or genetic disease. But the majority of patients with DCM and with HCM, as well as other inherited cardiomyopathies, are living without a genetic diagnosis, about 50% for HCM and as many as 75% for patients with arrhythmogenic cardiomyopathies and dilated cardiomyopathies. But the important thing to note is that even without that genetic diagnosis, family members can still be screened and should be screened with our clinical screening modalities like echocardiograms, ECGs, rhythm monitoring, etc. This is going to allow doctors to identify disease in patients who have it with or without knowing exactly where in their genome to look for that misspelling that puts them at risk. Additionally, there are a lot of efforts ongoing to improve genetic diagnosis. Many of these are centered around resolving variants of uncertain significance or those misspellings in genes that we don't know the consequence of. And additionally, looking with families with DCM and HCM to understand what the driving factors are for developing disease even without a genetic diagnosis. When we look forward to therapeutics, gene therapy and gene editing are being focused now at broader populations that include those patients who don't have a precision genetic diagnosis. So as we move forward as patients with gene-elusive cardiomyopathies, the future remains bright. It's important for us to know that many people across the world are working on improving the capability of genetic diagnosis for you and your families, and also that we can devise new therapies that use the most cutting-edge technology, even if we don't have a genetic diagnosis. Thank you so much.
This video is part of a series of educational videos developed by the Genetic Cardiomyopathy Awareness Consortium and brought to you by the DCM Foundation. Our goal is to connect you with the leading experts in the field of cardiomyopathy to further your understanding of this disease and to provide resources to help you manage your care. Thanks for watching.